Good day to everyone joining us, and welcome to today's Eurofins Panlabs webinar. Today's talk is entitled Predictive Assays for Cardiotoxicity using Human Embryonic Stem Cell-Derived Cardiomyocytes. My name is Mike Futala, and I'll be your host today. Today's webinar will run for approximately 60 minutes and includes a question and answer session. Now, this webinar is designed to be interactive, so please feel free to submit your questions and comments for our speakers throughout the presentation using the question chat box, and we will attend to your comments and questions during the Q&A session. This chat box is located in the control panel on the right side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please also note that this event will be recorded and made available to you on Eurofins Panlabs' website for future review. So a brief history about the company. Eurofins Panlabs Incorporated is a global CRO specializing in discovery, pharmacology testing services for over 40 years. Eurofins Panlabs' mission is to provide pharmacological testing services that predict clinical effects, and Eurofins Panlabs' scientific portfolio consists of over 1,800 assays, ranging from molecular assays to cell-based models through proof of concept in vivo activity determination. So now I'd like to introduce the speakers for today's event. They are Dr. Ying Zhao and Karen Marco. Dr. Zhao is a principal scientist and group leader of toxicology at Eurofins Panlabs. He is responsible for all activities related to the in vitro toxicology services, which includes cardiac toxicity, cytotoxicity, and genotoxicity assays. He serves as a director of client studies, coordinator of R&D projects, and manages new assay development and validation of ion channels, transporters, and CYP induction. Karen Marco is a senior scientist responsible for all activities pertaining to HCA functional toxicology, in vitro cellular assay development, and client services. She has extensive experience in molecular pharmacology assay development and automated high-throughput screening to develop, validate, and optimize cellular assays for early safety testing. She leads and manages HCA assay development and high-throughput screening projects with collaborative partners and clients, and also serves as study director for HCA functional toxicology client services. So now I'd like to hand over the presentation to Dr. Zhao. Please remember to chat in your questions in the chat box, and we will address them in the question and answer session following the presentation. Dr. Zhao?
All right. Thank you, Mike. Today we are going to talk about the assays that we developed using human embryonic stem cell-derived cardiomyocytes to predict cardiac toxicity. And this is the outline of our talk today. First, I will give you a brief introduction on the background of drug-induced cardiac toxicity and the assays that are recommended by regulatory agencies to assess the cardiotoxicity. I will also talk about the human embryonic stem cell-derived cardiomyocytes used in our assay. Then Karen and I will introduce you the 3 assays that we developed using human embryonic stem cell-derived cardiomyocytes by 3 technologies. We use multi-electrode array, the MEA, to measure cardiac field potential. We use the conventional patch clamp to measure the cardiac action potential, as well as image-based high-content analysis HCA to assess the cardiostructure toxicity. Why is early safety screening important? Drug discovery timeline is going longer, and the cost of the drug discovery is increasing. This figure here shows you the timeline and required process for a new drug development. According to Pharmaceutical Research and Manufacturer of America, it takes about 10 to 15 years to develop one new drug from the beginning to the end. The average cost is increasing over the years, as shown in this figure here. Now it is estimated to be between $800 million to $1 billion to develop a new drug. But even after the drugs got approved and on the market, there are a significant number of the marketed drugs being withdrawn due to toxicities that were not detected during the development stage. It causes tremendous economic loss as well as raising the patient safety concerns of the new drugs. And this chart here shows the major causes for a marketed drug to be withdrawn since 1970s. As we can see here, the cardiotoxicity, which is shown in red here, is one of the major reasons. Those are some examples in this table of non-cardiac drugs that have been removed from the market since 1990s because of their cardiac toxicity. Therefore, by identifying a cardiac liability early in the drug discovery can save money and time. And the cardiac toxicity of the drugs that were removed from the market was linked to a fatal electrocardiogram abnormality known as tarsal-depend, the TDP, which is caused by prolongation of the QT interval in ECG. And this is an electrocardiogram recorded from human. If we look at each unit, we can see that there are 5 major waveforms, P, Q, R, S, and T. So the interval between the beginning of the QRS complex to the end of the T waves represents the ventricular depolarization and repolarization. When the QT interval is prolonged, there is an increased risk of ventricular tachyarrhythmia, including tarsal-depend. If we record the electric activity at the cellular level by insert an electrode into a single cardiomyocyte, we can record a waveform showing here. This is the action potential. So the action potential corresponds to each waveform on the ECG. And there are 4 phases of the action potential. And different ion channels contribute to the different phases of the action potential. For example, the phase 0 is the rapid depolarization of the cell membrane. It's mediated by the voltage-gated sodium channel NAV 1.5. When the cell is resting, the intracellular is more negative than the extracellular. This stage is called polarization of the membrane. When the sodium channel opens, sodium flow from the outside the cell to inside the cell. It carries a positive charge, so it's depolarized the membrane. So the phase 1 is a rapid repolarization. It mediated by 2 potassium channels, Kv 4.3 and Kv 1.4. As shown in this cartoon, when the potassium channel opens, potassium flow from inside the cell to outside the cell is repolarized the membrane. And phase 2 is the plateau stage of the action potential. It's mediated by 2 ion channels. One is the calcium channel KAV 1.2 and the other one is potassium channel 1.5. So when the calcium channel opens, calcium flow from outside the cell to inside the cell. It counteracts the action of the potassium channel. So that's why we see a slowdown of the repolarization on this stage. The phase 3 is the repolarization phase, but it's mediated by the famous Hooke channel and the KV-LQT. Both of them are potassium channel. And the phase 4 is the resting phase. It's mediated by a potassium channel, the Kerr channel, that maintains the resting membrane potential of the cardiomyocytes. So interruption of any of the ion channel will have impact on the action potential and adverse effect will occur. And to address the cardiac safety issue, the International Conference on Harmonization, SAH, published a guideline on preclinical assessment in SAH S7b in May 2005. SAH S7b recommends 4 non-clinical approaches. First, determine if the compound belongs to a chemical or pharmacological class in which some members have been shown to induce QT prolongation in humans. For example, the drugs that are antipsychotics or histamine H1 receptor antagonists are likely to have cardiac safety issues. Then test the compound in an assay that measures the ionic current namely the patch clamp assays to determine the interaction of the compound with the ion channel, with the ion channels that are involved in the generation and propagation of the action potential. The third approach is the ECG recording from animals to detect the QT prolongation. The fourth approach is the action potential recording from cardiac preparations. And currently, hERG screening is the major assay used in cardiotoxicity assessment because all the drugs withdrawn from the market that cause the TDP blocks hERG channels. However, hERG channel assay is not perfect because not all compounds that inhibit the hERG channel result in QT prolongation. So in this case, the hERG assay produces false positive results. Moreover, arrhythmias can be induced by drugs that do not inhibit the hERG channels. In this case, the hERG assay produces false negative results. Here are some examples. The predictive value of the hERG assay is limited if a compound interacts with multiple ion channels. Verapamil is a classical example. It blocks both hERG and calcium channels, and it does not induce TDP clinically. There are some examples that compounds interact with other ion channels, such as NaV 1.5, but not hERG, can cause arrhythmias. So the hERG assay will not identify compounds that interact with kinase, GPCR, transporter, etc., which involve in the cardiac activity. The false results also come from species variation. For example, the standard action potential measurements are from either dog or rabbit cardiac preparation. It has been reported that dog and rabbit Purkinje fiber assays show less sensitivity to certain hERG blockers, such as Quinidine. The patch clamp assay cannot detect compounds that cause structural damage on the heart. To solve those problems, different screening strategies are developed. For example, there are more ion channels such as NaV 1.5, CaV 1.2, KVLQT, etc. are screened. However, it adds the cost and delays. And action potential measurement is another approach. The action potential is synchronized action of all the ion channels involved. However, it is low throughput and labor intensive. Moreover, it has species variation due to the difficulty to use human preparation. Another solution is to measure the extracellular field potentials from cultured cardiomyocytes by multi-electrode array. The field potential duration measured by MEA correlates closely with the QT interval in the electrocardiogram. When used the stem cell-derived cardiomyocytes, it solves the species variation problem. So the image-based high-content analysis can detect compounds that cause structural damage. In our assay, we use the human embryonic stem cell-derived cardiomyocytes, which are commercially available from GE Healthcare in UK. GE Healthcare did a thorough validation on the cells in terms of morphology and function. And this picture shows you the characteristic staining of the GE Sativa Plus and the cardiomyocytes. And this picture here shows you a beating, shows you a monolayer of the beating cells. So those cells are human origin. They are functional verified. They possess all major ion channels. There are representative myocytes population in the culture. Majorities are ventricular cells with some population of atrial and nodal cells. And they are karyotype verified, renewable, reproducible, scalable, therefore suitable for high throughput assays. Excuse me. Using GE Healthcare's human embryonic stem cell derived cardiomyocytes, we developed 3 assays to assess compounds' cardiac liability. We used the image-based high-content analysis shown here to detect the compounds that cause cardiac structural damages. We used the multi-electrode array MEA method to detect the compounds that cause and to measure the field potential parameters. We use the conventional patch clamp to measure the action potential from the cardiomyocytes. So our assays provide a comprehensive assessment of both functional and cardiotoxicity by MEA and conventional patch clamp, as well as structural cardiotoxicity by high-content analysis. Using human embryonic stem cell derived cardiomyocytes, we provide a biological relevant alternative to current cell models and primary cells to predict cardiotoxicity. Those three assays can be performed as a package to give you a comprehensive prediction of your compound cardioliability. Also, you can choose individual assays to address certain concerns. The human origin of the cells avoids species variation, so they are cost-effective assays when compared with the assays using animal. Those assays are fully validated. They provide reliable and reproducible data. We use the MEA instrument from Axion Biosystems. The signal can be recorded from 1, 12, 48, or 96 culture wells. The cells are cultured on the MEA plate, which have multiple electrodes embedded on the surface of the well bottom, as shown in this picture. Those are the electrodes in the MEA plate. You can see there's monolayer of cells cultured on top of the electrode. So those electrodes can pick up the extracellular electrical signal from those cells. The key features of the MEA is showing here. The assay is high throughput, which use up to 96 well plates. It provides rich information. It records ECG lag traces. We can measure different parameters at the same time, such as the field potential duration, the beat rate, amplitude, slope, and conduction, velocity, etc. The MEA assay is a non-disruptive label-free technology. Therefore, it preserves cell function and the cellular connectivity, which is important for this application. Because of this, we can do recording to analyze the long-term effect of the compound over a few days. We also can analyze the action potential propagation properties. It is easy to use and requires less skill compared to manual patch clamp. And this slide shows you the protocol we use for the MEA assay. And the panel here shows the cell culture protocol. So we saw the cell directly from the cryo preserved the wells, and we see the cell directly onto the MEA plate, which coated with a fibronectin. The cell is cultured for 4 days at 37 degree 5% CO2. On day 4, we change the medium. It's 1 day before the recording. On day 5, we perform the MEA experiment. The panel here shows you the MEA experiment protocol, and we test the 5 concentrations on each well. So we test the wheel control first, and the cell incubated with the wheel control for 15 minutes, and then we recorded the extracellular field potential at the last 5 minutes. And after the recording, we add the lowest concentration, and then it's incubated with cell for another 15 minutes. We recorded for the last 5 minutes. So the remaining concentration, we use the same protocol to record it, all the effect. We measure 3 parameters from the MEA: the sodium spike amplitude, inter-spike pair rate, and field potential duration. So this is one example recording traces here. The spike here is called sodium spike. It's mediated by the opening of the sodium channel. So the interval between the sodium spike and the wave right here is the field potential duration. It is the analog of the QT in the ECG, and the interval between the 2 sodium spike is the beat pair rate, also called inter-spike pair rate. And this is one example showing the hERG channel blocker E4031 that prolongs the field potential duration of the cardiomyocytes. So this recording on top is the control recording. As we can see, this is 1 sodium spike, the second, the third, and then this duration here is the field potential duration. And after we apply the 0.1 micromolar of E4031. So you can see visually here the field potential duration is increased compared with the control. If we overlay the traces of different concentration of E4031, we can have this this plot. The blue trace is here is the control, and the green one right here is the 10 nano. It's the it is 1 nanomolar of E4031, and then this is 10. This is 100 micromolar of E4031. We can see the field potential duration is prolonged following the increase of the concentration of the E4031. And this is another example that shows the this is the analyze the result from the E4031. As you can see that the E4031 significantly increased the field potential duration at the concentration of 10 nanomolar, and it did not increase the change the interspec interval and the spec amplitude up to 100 nanomolar. This is the analyzed data from a calcium channel blocker, which is nifedipine. We can see the nifedipine decrease the field potential duration and also decrease the interspec intervals. And so this slide here shows you the summary results from the MEA. We test the E4031 as I explained to you. It's the hERG channel, the blocker. As we can see here, the action potential, the hERG channel contribute to the repolarization of the action potential. When the hERG channel is blocked, the repolarization of the action potential is delayed. You will see a prolongation of the action potential duration. As expected, we see the increase of the field potential duration. So nifedipine is the calcium channel blocker. So on this plot, we see that the calcium channel contribute to the plateau of the action potential. When it is blocked, the action potential it's going to increase the rate of repolarization of the action potential. As we see here, the field potential duration is decreased. It's correctly showing the result as the mode of action of this compound. So we also test the Quinidine, which is the sodium channel blocker. And so when you block the sodium channel, if you see from this plot, the uprest of the action potential is mediated by the sodium, so by the sodium channel. If the Quinidine is a sodium channel blocker, so we see the reduction of the spec amplitude, which is a representative of the uprest of the action potential. So, and also we see the increase of the field potential duration because the Quinidine is also blocks the hERG channel at higher concentrations. And we also test the IKS blocker as a chromolar 293B. So in this case, we did not see any effect up to 3 micromolar. So we were not able to go higher than 3 micromolar because the cells stop beating at 3 micromolar. So we will not be able to detect the effect on those parameters at a higher concentration for this compound. So we test Sotalol, which is a beta-adrenergic antagonist, and it is also a hERG channel blocker. So we see increased the field potential duration in this case at 1 micromolar. So another one is the isoproterenol. It is a beta-adrenergic agonist. So in this case, we see a decreased interspec interval and a decreased field potential duration. So the results are similar with the results that observed from other assays for those compounds. This slide shows you the action potential recording by the conventional patch clamp. This is the protocol we use. So we use the perforated patch clamp technique. The assay was performed under the physiological temperature. And we measure the resting potential, the amplitude of action potential, the maximum rate of rest of the upstroke of action potential, the action potential duration at 60% and 90% of repolarization. And We perform the statistic analysis and report as a statistical significant difference when the p-value is less than 0.05. And this is one example recording from Quinidine. As you can see, the action potential duration recording, this one is the control. Those are the traces after treatment of Quinidine. For example, this one is at 0.3 micromolar, this is 1 micromolar, this is 3 micromolar. So we observe that at higher concentration, 3 micromolar, all parameters show significant changes. So the action potential duration at 90% of repolarization is the most sensitive parameter in this case. This is the summary result for the action potential experiments we performed. We tested 3 compound sotalol, which is a hERG channel blocker, also a beta adrenergic antagonist, and also the quinidine, nifedipine. So we see those characteristic effect of those compounds. So those results we obtained are comparable to the published results from the animal cardiac preparation, or either iPS or embryonic stem cell derived human cardiomyocytes. So I talked about the assay stats from the MEA and the conventional patch clamp. Those measures the functional cardiotoxicity. Now I'm going to transfer the microphone to Karen. She's going to talk about the high content analysis experiment.
Thank you, Yu. My presentation today will cover the utility of using image-based high content analysis for the development of predictive cardiotoxicity assays using human embryonic stem cell derived cardiomyocytes. Improving cardiac drug liability surveillance requires predictive in vitro assays that determine drug impact on electrophysiological, structural, and functional components of cardiomyocyte biology. Human embryonic stem cell derived cardiomyocytes provide a single integrating cell model compatible with a wide range of analytical platforms for thorough analysis of drug liability. Cardiotoxicity profiling using human embryonic derived stem cells cardiomyocytes allows complementary multiparametric analysis of both electrophysiological and structural responses to drugs, as well as assessment of a wide range of cardiac liabilities and cardiotoxicity mechanisms. High-content assays are useful for identifying structural cardiotoxicity caused by drugs. The data generated complements those from electrophysiological recordings by identifying compound liabilities due to morphological damage or loss of cardiomyocyte viability. Many kinase inhibitors show cardiotoxicity even though they do not bind to ion channels. To provide comprehensive early safety screening, assessment of both functional and structural cardiotoxicity is essential for discovery of safe drugs. We have used GE Healthcare Sativa Plus cardiomyocytes in combination with our high-throughput screening platform to validate in vitro high-content assays that evaluate the potential for drug-induced structural cardiotoxicity. These assays are provided in a 384-well format with automated liquid handling and compound transfer, quantitative multiparametric image analysis, and automated data reduction and report generation. Our cell health assay measures key indicators of calcium homeostasis and mitochondrial and cell membrane integrity. Our cardiotox assay includes parameters for assessing cardiomyocyte alpha-actinin and troponin integrity and perinuclear B-type natriuretic peptide protein expression. The GE Healthcare Sativa Plus cardiomyocytes are derived from an NIH-approved stem cell line, have been extensively characterized and functionally verified for cardiac transcription factor expression, structural markers, and individual ion channel activity, and supply a renewable, reproducible, scalable cell source in a cryopreserved, ready-to-use format. For the HCA assay protocol, cells are thawed and seeded at 9,000 cells per well in 384-well fibronectin-coated microplates and RPMI 1640 medium supplemented with B27. Medium is exchanged on day 4 and on day 7 prior to compound addition. 24 and 72-hour compound incubation time points are tested. The 4-color readout generates a multiparametric data set. Events are imaged at the subcellular level to understand perturbations associated with non-ion channel drug liabilities and to provide information on mode of action of cardiotoxic drugs at the cellular level. These HCA assays were developed and validated with 12 reference compounds shown in this table. Classification for structural and non-structural cardiotoxins was obtained from FDA approval package cardiac warnings and literature references. Drugs were selected with different primary pharmacology and 1 hepatotoxin was included. Assay positive control compounds were selected based on ability to induce significant biomarker induction at both assay time points. Amiodarone was selected as the positive control for the live cell health assay, and nifedipine used as negative control to generate cell health profile plots. Mitoxantrone was selected as the positive control for the fixed cardiotox assay. For the live cell health assay, cells are treated with a 7-point dilution series of test compound for 24 and 72 hours. Standard final assay top concentration for testing is 100 micromole. Cells are stained with multiplex dyes, HEST, Flow4AM, TMRM, and Toto-3 iodide. Automated fluorescent microscopy is carried out using an ImageXpress Micro XL wide field imaging system and MetaXpress image acquisition analysis software from molecular devices. Images are collected with a 20x objective with 4 fields of view in each of 3 replicate wells for each concentration. Assay deliverables include cell count, EC50 and IC50 curves, which are based on nuclear staining with HES. Calcium mobilization is detected by Flow4AM staining with significant response defined at 3.5-fold and 6 standard deviations above vehicle background. Mitochondrial membrane potential is determined by TMRM staining with significant response defined at 2-fold and 6 standard deviation change over vehicle background. Membrane permeability is detected with Toto-3 staining with significant response defined at 5-fold and 6 standard deviations above vehicle background. Secondary data visualization cell health profile plots with the 6 parameters shown are plotted at compound dilution concentrations above and below the IC50 value. Customized tools are used for quantitative imaging, data analysis, and data visualization. For the cell health assay image analysis, nuclei are segmented by the HES staining. A dilated collar around the nuclear region is used to sample flow-4 intensity for detection of free calcium. Mitochondrial organelles are segmented by the TMRM staining in the cytoplasmic region around each nuclear region. Viable and non-viable cells are identified on the basis of Toto-3 intensity. The AIM data output is normalized to the number of cells in the well and vehicle wells. Relative cell count POC is equal to a ratio of the fluorescent integrated intensity of treated wells to the integrated intensity from the vehicle wells. For each biomarker fold induction, the integrated intensity is first normalized to the nuclear integrated intensity in their prospective wells, and then this ratio of each well is normalized to the vehicle wells. The AIM cell health profile plot data is normalized to % maximum response, and the data output is scaled 0 to 1. The 6-parameter profile plots provide information regarding cell viability, mitochondrial status, and nuclear and cellular calcium. Plots charted above and below the IC50 value allow visualization of mechanistic indications of toxicity. Significant change is defined as parameters demonstrating greater than 6 standard deviation change over mean vehicle background. The live cell health assay data format reports EC50 and IC50 values the concentration of significant biomarker fold induction and the maximum fold induction of each biomarker. The cell health assay positive and negative control validation is summarized in this table. The 6-parameter cell health profile plots are shown overlaid for the positive control amiodarone at 31.6 micromole, traced by the blue line. The negative control nifedipine at 31.6 micromole, traced by the red line, and the vehicle 0.5% DMSO, traced by the black line. The negative control nifedipine and vehicle plots, as you can see, overlay one another quite closely, indicating no drug effect detected. Compound-treated profile plots can be overlaid with vehicle plot profiles, allowing a direct comparison of treated and non-treated responses. The arrows on the profile plot indicate amiodarone effect on each of these parameters. We see a decrease of mitochondrial membrane integrity, an increase in the mean form factor indicating cell shape is rounding and cells are dying, increased calcium mobilization, and decreased viability. This is an image showing the toxic effects of sunitinib treatment on cardiomyocytes. The nuclei are stained blue with Hess. The accumulating free calcium is stained with flow4 in the green, and the remaining mitochondria is stained with TMRM, and you can see it with the red. The assay results for sunitinib treatment. These are representative graphs for IC50 curve and biomarker fold induction, as well as the tabular report format for the validation data for cell parameters for sunitinib treatment in the table below. The 6-parameter cell health profile plot for 24-hour sunitinib treatment are shown. Compound concentrations are plotted in green, above, below, and at the IC50 value, and overlaid with the positive, negative, and vehicle control plots. The data table summarizes significant parameter changes. 0 indicates no change, plus significant increase, and minus significant decrease. Blue highlights on the table indicate that the parameter demonstrated greater than 6 standard deviations change over mean vehicle background. Increasing concentrations of sunitinib treatment cause decreasing cell viability. The cell health profile plot data identifies the parameters that are impacted as the level of toxicity increases. This information provides concentration-dependent mechanistic insight for identifying the cause of toxicity. For the Cardiotox fixed assay, cells are treated with a 7-point dilution series of test compound for 24 and 72 hours. Standard final assay top concentration for testing is 100 micromole. Cells are fixed and stained with DAPI, anti-alpha-actininin, anti-troponin-1, and anti-BMP32 peptide. Automated fluorescent microscopy, again, is carried out using an ImageXpress Micro XL wide field imaging system and MetaXpress image acquisition and analysis software for molecular devices. Images are collected with a 20x objective with 4 fields of view in each of 3 replicate wells for each concentration. Assay deliverables include cell count EC50, IC50 curves based on the DAPI staining. Alpha-actininin sarcomere disruption is detected by the anti-alpha-actininin labeling. Troponin-1 integrity loss is detected by the anti-troponin-1 labeling. Perinuclear BMP protein expression is detected by the anti-BMP32 labeling. For all biomarker parameters in this assay, significant response is defined as 5-fold and 6 standard deviations above vehicle background. Image analysis for the fixed cardiotox assay includes masking signal for drug-induced changes in distribution of alpha-actininin protein before and after treatment. Alpha-actinin is a protein involved in formation and maintenance of Z-lines of cardiac myophils. Troponin-1 protein is a regulatory protein in cardiac muscle contraction and relaxation and a marker of myocardial damage. And the third component of this assay is an expression of B-type natriuretic peptide, a neurohormone synthesized in cardiac ventricles in response to wall stress. During secretion from cardiomyocytes, proBNP is converted to a physiologically active 32 amino acid C-terminal BNP fragment and an inactive N-terminal fragment NT-proBNP. Accumulation of BNP32 in the perinuclear region of cardiomyocytes is indicative of cell stress. For image analysis, the nuclei are segmented by DAPI staining. Alpha-actinin signal is sampled in the cytoplasmic region and is defined by vesicle area and intensity thresholds for alpha-actinin labeling of the sarcomere disruption, which is evident in the image that I have the cursor on. Troponin-1 is sampled in the cytoplasmic region and is defined by vessel area and intensity thresholds for anti-troponin-1 labeling of changes in troponin integrity. You can see that after the mitoxantrone treatment here. BNP32 is sampled in the cytoplasmic region and is defined by vessel area and intensity threshold for anti-BNP labeling and sampled in the perinuclear region. Fixed assay data format reports EC50 and IC50 data, the concentration of significant biomarker fold induction, and the maximum fold induction for each biomarker. The fixed assay positive control mitoxantrone validation is summarized in this table. Biomarker data is normalized to the number of cells in the well and vehicle wells. Note the biomarker signal for each of the parameters represented by these red traces increases with decreasing cell number as a result of toxicity produced by the mitoxantrone treatment. These images show the toxic effects of sorafenib after 72 hours of treatment. The nuclei stain blue. Sarcomere disruption is detected by the bright green to yellow punctate vesicle anti-actinin immunolabeling, and changes in troponin integrity are detected by the bright red vesicle anti-troponin immunolabeling. Perinuclear BNP protein expression is detected by the uniform orange vesicle anti-BNP immunolabeling. These are the assay results for sorafenib, representative graphs for IC50 curve, and biomarker fold induction, as well as the tabular report format for the validation for cardiotox parameters for sorafenib treatment are shown. HCA early safety screening workflow employs GE Healthcare Sativa Plus cardiomyocytes to test small molecules and biologics in a high-throughput 384-well format. A state-of-the-art imaging platform using molecular devices, Image Express Micro XL, Meta Express image acquisition and analysis software to generate the 4-color readout that produces multiparametric data sets. The data is analyzed and QC'd. Each curve is QC'd for best fit reference compound EC50, IC50 with within threefold of historical average. The multiparametric informative data generated results in early identification of drug toxicity liabilities. Why Eurofins Panlabs for early safety cardiotoxicity screening? Our new biologically relevant cell model, GE Healthcare Sativa plus Cardiomyocytes, combined with image-based HCA in vitro cardiotoxicity testing, generates data results with mechanistic insight that aid in informed decision. The HCA data provided complements those from our electrophysiological recordings by identifying compound liabilities due to morphological damage or loss of cardiomyocyte viability. Our assays offer a cost-effective, comprehensive predictive assessment of both functional and structural cardiotoxicity in a single cell line model. The service is provided by an excellent scientific team taking meticulous care to follow quality control steps to ensure delivery of high-quality informative data. For more information regarding these assays and scheduling, contact our customer support team. Jung and I would like to acknowledge our colleagues for their contributions to the work presented today. Jessica Chestnut-Spielman for many contributions to the HCA assay development and validation work, Tom Arnold for his IT support in automating data reduction and report generation for the HCA and EFIS work, Shimin Wang and John Kushlecki for their contributions to the electrophysiology assay development and validation, and our colleagues at GE Healthcare, Jan Turner for coordinating this collaborative project across the miles, Mike Clements for his assistance with the EFIS work, Liz Roquemore, Nick Thomas, Ray Ismail, and Sun Young for their assistance with the HCA work.
Great. Thank you, Dr. Zhao and Karen, for this very interesting and informative presentation. So now we can begin our Q&A session, and I would like to invite the audience to continue sending in your questions and comments right now using the question window for this Q&A portion of the webinar. So we've received several questions, and we're going to take about 20 seconds to review real quick, and then we'll be right back in about 20 seconds. Thank you very much for your patience. All right, thank you so much for your patience. So the first question is, do you recommend we do both cardiomyocyte assays as well as HERG assays?
And I'll answer this question. So we do recommend to do the both assays. The HERG screening assay is still a very valuable assay. It's a relatively high throughput, and the cost is relatively low. And as it has been already on the market for many years, a lot of validation study out there, and the product value of HERG screening assay is still relatively high, even though with some limitation on that. So the cardiomyocytes work, in my opinion, is going to fill the gap between the HERG assay and the QT prolongation. So it's going to add value to the current existing assays.
Okay, thank you very much. And the next question, how does the cost of Sativa Plus assays compare to HERG assays?
The cardiomyocyte assays is going to be more expensive than the HERG automated patch clamp assays, and this is because the cost of the cell itself is significantly higher than the HERG screening assay, and also for the MEA assay, the cost of the plate is relatively high, and also there's not too much automation you can do even with the high throughput assays, so the labor cost is relatively high. So it's going to be higher than the automated patch clamp, but I think it's comparable with the manual patch clamp assays.
Okay, thank you. So the next one, what is the IC50 value for E4031 using HESC-derived cardiomyocytes, and how does it compare with the animal models or human trials?
For the MEA work we did on the with the E4031, so we did not generate IC50 value. The challenge for that is when the concentration go at higher concentrations, the cells stop beating. So in order to measure the endpoints, we need the cell to beat. If the cell stop beats, then there is no measurement at all. So in order to fit the IC50 value, you need to reach a plateau. You need to know the maximum effect, either the inhibition or induction. And if you don't have the maximum value, it's very difficult to fit the IC50 value. So we do not have IC50 value in this case. And the way so we can use the data is by looking at the statistically significant difference of the compound treatment group compared with the control group. When the difference is significant, usually that's the point that considering there's a toxic effect.
Okay, thank you. And we have time for one more. What is the benefit of running the 2 time points in the HCA assay?
There is substantial benefit for running both time points in these assays. Not all compound toxicity appears in a 24-hour incubation time frame. Some of the parameters spike early and drop off, and others take a while to present themselves. So running 2 time points in a toxicity assay is basically you're not letting anything slip by, and you're giving a more thorough analysis of the toxic liabilities of the compounds.
Okay, great. Thank you for those answers and the presentation as well. And so we've now reached the end of the question and answer portion of the webinar, and I'd like to direct you to, if you have further questions, please direct them to Dr. Zhao's and Karen Marco's email address showing on your screen. So thank you everyone for participating in today's webinar, and you'll be receiving a follow-up email from Eurofins Panlabs with access to the recorded archive of this event. Also, as you leave, a survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our future webinars. So now please join us in thanking our speakers, Dr. Zhao and Karen Marco. We hope you found this conference informative, and have a great day, everyone. Thank you.